Breakthrough. Nanosize device uncloaks cancer cells in mice and reveals them to the immune system. I'm Steve Belair and welcome to Breakthroughs. Scientists at Johns Hopkins report that they have designed and successfully tested an experimental super small package able to deliver molecular signals that tag implanted human cancer cells in mice and make them visible for destruction by the animal's immune system. The new method was developed, says the researchers, to deliver an immune system uncloaking device directly to cancer cells. Conventional immune therapies generally focus on manipulating patients' immune system cells to boost their cancer-killing properties or injecting drugs that do the same but often have toxic side effects. Results of the proof-of-concept experiments were published on February 7th in the Proceedings of the National Academy of Sciences. The hallmark of cancer biology is a tumor cell's ability to essentially hide from the immune system cells whose job it is to identify and destroy cancer cells. Current cellular immunotherapies, notably CAR-T, require scientists to chemically alter and enhance a patient's own harvested immune system T cells. An expensive and time-consuming process, says the researchers. Other weapons of the arsenal of immunotherapies are drugs, including the so-called checkpoint inhibitors, which have broad effects and often lead to unwanted immune system-associated side effects, including damage to normal tissue. By contrast, the Johns Hopkins team sought an immune system therapy that can work like a drug, but also individually engineers a tumor and its surrounding environment to draw the immune system cells to it, says Jordan Green, PhD. And our process happens entirely within the body, Green says, requiring no external manipulation of the patient's cells. To develop the new system, Green and his team took advantage of the cancer cell's tendency to internalize molecules from its surrounding. Cancer cells may be easier to directly genetically manipulate because their DNA has gone haywire. They divide rapidly and they don't have the typical checks and balances of normal cells, says Green. The team created a polymer-based nanoparticle, a tiny case that slips inside cells. They guided the nanoparticles into cancer cells by injecting them directly into animals' tumors. The nanoparticle method we developed is widely applicable to many solid tumors despite their variability on an individual and tumor type level, says Green. Once inside the cell, the water-soluble nanoparticle slowly degrades over a day. It contains a ring of DNA called a plasmid, but does not integrate into the genome and is eventually degraded as the cancer cell divides, but it stays active long enough to alter protein production in the cell. The additional genomic material from the plasmid makes the tumor cells produce surface proteins called 4-1-BBL, which work like red flags to say, "I am a cancer cell, activate defenses." The plasmid also forces the cancer cells to secrete chemicals called interleukins into the spaces around the cells. The 4-1-BBL tags and interleukins are like magnets to immune system cells, and they seek to kill the foreign-looking cancer cells. Essentially, we're forcing the tumor to open itself up and instruct immune cells to kill it, says Sang. In their animal experiments, Sang and the Johns Hopkins teams injected the loaded nanoparticles into tumors created by implanting mice with either human melanoma or colon cancer cells. A control group of mice implanted with melanoma cells receives systemically an immunotherapy drug known as anti-PD-1 antibody. All of those mice died quickly, within 2.5 to 3 weeks due to tumor growth. Then the research team injected other groups of mice, which were also injected with cancer cells, with nanoparticles containing only one or both of the uncloaking signals, the genetically encoded 4-1-BBL tags and the interleukins. In mice with implanted melanomas, the nanoparticles that combined the two signals had a stronger effect than either signal alone. The medium or midpoint survival of the mice with the combo signal package was 40 days, and about 20% of them lived through the end of the 60-day study period. The researchers also saw that some of the mice in the treated melanoma group developed vitiligo, a condition in which skin cells lose their pigment. It occurs in humans too, including in people undergoing immunotherapy for melanoma. It's generally thought that vitiligo in melanoma patients is a sign that the immunotherapy treatment is working, and the immunotherapy is spreading to other parts of the body where other melanocytes reside, said Sang. The tumors shrank away in all of the mice with implanted colon tumors that received the nanoparticles with both signals, and they survived through the entire 60-day study period. When the researchers re-injected human colon cancer into the sides of the mouse opposite of the original tumors, unlike with age-matched controls, the newly implanted cancer cells failed to form a tumor, suggesting a lasting effect of the boosted immune system. Wow, I'll leave a link in the description to the full article, and as usual, please like, share, subscribe, and take care.